So we are truly in the very end of the semester, right? We only have 2 more lectures for this class. So what I'm going to be doing for these 2 last lectures is we're going to be covering some topics of sort of where control of insects may fail, so talking specifically about resistance to control techniques. And then on Wednesday, we'll be talking about what are the things we can do to combat resistance and failure of control, things like that. So just some reminders before we get started. There is no lab this week because obviously we don't have a Friday class. It's a dead day, so there's no class then, so we can't have other labs. However, the lab room will be open for the next 3 days for microscope use, since, as I'm sure many of you who have just recently started identifying your insects may realize, I sort of sat there and watched the sheer lack of students coming into the lab to use microscopes and thought, I'm going to be swamped for the 2 days before the collection is due. Because the reality is that there are a lot of insects you can identify to family very easily. What you're probably going to find is there will be 5 or 6 insects, maybe more in your collection, that are going to be ridiculously difficult to identify to family without a microscope. And so basically today, from the normal lab time, I'm going to have the lab open. I'm not going to be there for the first hour. I've got a meeting, but I've got a grad student coming in, Rohit. He'll be watching. And so you just come into the lab, ask him, and he'll unlock a microscope for you. Tomorrow, we'll have the lab open almost all day, from 9 to 5, with a 1-hour gap. There's a class at 11 o'clock. I think it's a bit class is in there. So we won't be available then. And then on Wednesday, from 12 to 5, the lab will be open again. If you want to use microscopes during those times, I'll probably just be in my office. So knock on the door or walk in the lab and shout through the closet room. And I can come out and get you a microscope. So yeah, take advantage. Microscopes are handy. And a reminder, the collection is due this Wednesday at 5 p.m. in the room 119. So you can either stop by my office or take it to 119. Main point is, let me know when you turn it in so I can put them somewhere where they won't get bothered by people, right? Other than that, the final is this upcoming Monday. So it's right off the bat. It's at our usual time, 11 o'clock, all the way to 1 p.m. So you got 2 hours, and it's going to be here in this room as usual. So be ready for that. The final is what I call semi-cumulative, meaning the vast majority of material is going to be 3/4, probably more like 4/5 is going to be stuff we've covered since the last exam. There will be a little bit of material that'll be broad. It'll cover stuff covered in the first 2 exams. But I'm not asking for nitty-gritty stuff. What I'm asking for is broad concepts about entomology in those. So I'm not going to be asking you to name the parts of the legs or things like that. Okay? All right, so we do have an interesting insect today because I think this is an insect that relates really well to the topic of resistance that we'll be talking about, and that's the Colorado potato beetle. So this guy's coleopteran. It's in the family Chrysomelidae, which is the leaf beetles. So this is a big family of pest beetles. I imagine some of you have found some chrysomelids, and they are in your collection. I wouldn't be surprised. When I did my insect collection for grad school, we had a requirement. It was 72 insects, and they all had to be from different families. And so I remember collecting a whole bunch of beetles and being so pissed when like half of them turned out to be chrysomelids. But it's a big family. There's a lot of them. They're very charismatic. So these guys are native to the Rocky Mountains, and they were really fine. They weren't pests. In 1811, they were found feeding on wild solanaceae, which are the nightshade family, right? So potatoes, tomatoes, those sorts of things. But when they started growing potatoes in the United States, these guys host shifted. They moved from wild solanaceae over to potatoes, and they very quickly became the biggest insect pest of potatoes in the New World. The larvae basically emerge from the ground. They crawl up the plants. They feed on the leaves, and they can rapidly defoliate. Here we have some larvae. You can see they skeletonize the leaf. Kind of the lights turned off real quick. We can turn it back on in a second. There we go. And here are some potato plants that have been swept through by Colorado potato beetle larvae. And you can see there's actually really no leaves. It's all just stems. So they can wipe things out pretty quick. These guys spread. Within like 30 years, they moved from the Rockies all the way to the East Coast. By the 1920s, they were found in Europe. They're all over the place. So one reason we're particularly interested in these guys is they're considered one of the real bad actors as far as insecticide resistance is concerned. Our control for the beetle is almost entirely reliant on chemicals. And then on top of that, these guys basically feed on Solanaceae, which is arguably the most toxic family of plants on the planet. So they are used to getting exposed to all sorts of different toxic chemicals. So these beetles have top-notch detoxification enzymes. And so they are arguably the most chemically resistant insect. They're resistant to more than 52 active ingredients, 13 different modes of actions. Not necessarily any given beetle is resistant to all of these, but there have been examples of resistance to all of these, at least in single populations. And it got so bad to the point that in the 1990s, many growers actually ran out of chemistries that worked against Colorado potato beetles. So you'll notice in this chart, this is the year of the report of chemical resistance in the beetle. You see it takes off pretty sharply in the 60s, and then it kind of lulls in the 1990s. You see that the number of reports slows down. That's not because the beetles were susceptible to the chemicals, it's because they ran out of chemicals. There weren't any new reports to be filed. So it was a bad situation. Consequently, in 95, neonics came along, and so people thought, oh, this is great, but you can see in 2000, that's when the beetles developed resistance to neonics, so you see the sharp uptick in reports of resistance again, because they were overcoming the neonics. But on the other hand, I think these beetles are interesting from a historical perspective, in line with sort of Cold War and World War II propaganda. So these beetles come from the United States. They were introduced to Europe, and so there's a whole lot of people, there's a lot of conspiracy theories about how they got to Europe and the bad actions of the US government in supposedly trying to wipe out various European actors that they don't agree with. And so you get all of this sort of what they call, in Germany, the beetle was called the Amokoffer, which means the American beetle or the Yankee beetle, as they would call them commonly. The distinguishing feature of the beetle is these orange and black, or white and black stripes on the back. So you see lots of posters with the beetles with white with red stripes, with stars on their backs, things like that, to show that they are truly American beetles. But so during World War II, essentially there were these stories going around that what the American government was doing was they were flying planes over Germany and they were throwing these Colorado potato beetles out the back of the planes onto German potato fields to do some sort of biological warfare on the Germans. Consequently, there were rumors that the Germans were doing the same thing to the British Isles. And so there was a lot of sort of finger pointing and people saying somebody was doing this, somebody was doing that. In reality, there's no evidence that anybody was doing that except the Germans dropping potato beetles on themselves. Well, which was the Germans were actually experimenting with whether they could drop Colorado potato beetles in Britain. And so they bred this really hardy variety of Colorado potato beetle, and they designed these boxes that they could drop out of planes. So to test it, they flew these planes and they threw a bunch of the boxes out in Germany so they could throw them out and count the infestation levels and do a little scientific experiment. And I guess they decided it was too much effort and the amount of damage they caused wasn't enough to actually warrant using them as a form of warfare. But during the Cold War, it was believed that Americans were smuggling these beetles into West Berlin, right? So after World War II, Germany gets split in half. There's East Germany, which is ruled by the USSR. It's a communist government. There's West Germany, which is ruled by the USA. Well, not by the USA. It's a democratic society, but it's allied with the US. So you got this sort of division in the Cold War. But Germany's capital, Berlin, is firmly set in East Germany over where the USSR has sway. And so they split that city in half as well. And so there was this rumor that essentially they were smuggling in beetles into West Berlin and releasing them from there, releasing them from various military bases in the area to go and infest various Eastern Balkan states. And it got to the point that the Eastern German government actually released this pamphlet, Halt Amokoffer, with these lovely American beetles. on it. And it's kind of lovely. It's got these great illustrations of what you should do. So these would be like, there were these, yeah, with children with spirit, and by spirit they mean alcohol. So there were these stories of children would go to school, and immediately after school they'd have an hour of mandatory Colorado potato beetle picking. Because they're so resistant, the only way to control them was you would literally pick up the beetles and you drop them in alcohol. And so you just would go out and brute force it, just collect tons and tons of adults and drown them in alcohol or sudsy water. So you get some interesting stuff. You get these children collecting. You got a nice picture of the beetle. You got this weird illustration of beetles with like peasant garb with their hands tied behind their backs, being dropped in alcohol. It's just bizarre. But anyway, that's just sort of an aside. Consequently, Colorado potato beetles are still in the public eye in Europe, especially because in 2004, when there was Russia essentially taking Crimea, and the rebellion along the pro-Russian separatist right sort of rebelled and took control of the eastern border of Ukraine. And so these pro-Russian separatists got a nickname called the Coloradi, after the Colorado potato beetle. And it's because the separatists, their symbol was this orange and black striped St. George's ribbon right here, which kind of looks like the pattern on the back of the beetles. And so they were called the Coloradi after the beetles. There's all sorts of this is Putin here dumping a bucket of Coloradis over the border into Ukraine. So lots of symbolism there. So it's just it's an important beetle, especially in parts of the world where the potato is a real big part of the staple diet. So anyway, moving on from oddities in insect culture. Last week we were covering insect-specific approaches to pest control, talking about physiological behavioral manipulation. Now we're going to talk about resistance to pest control tactics. So how is it that pest control tactics work really well for a while, and then eventually they tend to fail, and we have to find new technologies to use against them? So again, as with lots of things in this course, a lot of it goes back to the 1940s with the introduction of synthetic organic insecticides. These guys were introduced, and people basically assumed that we were done dealing with insects. There were entomology classes where the professors would come in and say, "You are so lucky, you get to go out and collect insects because all these species are going to be extinct, in like 20 years. So this is going to be like the last collection that anybody ever has to do." And they were wrong. But consequently, people thought they were going to do really well and insects wouldn't be able to figure out a way to get past it. But by 1947, there was a population of houseflies in Sweden that were found to be unaffected by DDT. You could spray them and they'd have nearly 100% survival, right? And then they found many, many more reports of insecticides failing after that. And they ended up describing this as what they called resistance to insecticides. Resistance being a description for any decrease in susceptibility of a pest population that had previously been managed effectively with that technique. So essentially, you had a technique that worked on a specific population, like DDT on these houseflies in Sweden, and over time, that effect stops working. And that's how we define resistance broadly. And typically, we see that resistance is only relevant, or is at least only discussed to the extent that the technique is no longer effective or economical. You may see small increases in resistance where it's a little bit less effective, but because this is associated with economic entomology, it's an economic term. We're generally interested in this from the perspective of whether or not the loss of control is economic or not. So generally, this set up the real pattern for how effective insecticides were and how resistance established itself in the following years. Essentially, companies would develop a new mode of action or a new chemistry that played off of a mode of action differently. They would introduce it, and between 2 and 20 years later, they would have their first report of resistance. And, in 1979, this ultimately resulted in the United Nations reporting that resistance was one of the top 4 environmental problems that was facing the world. So at global scale, they saw resistance as a top 4 problem. Of course, it's only worse Now, if you look at the chart that we have over here, on the vertical axis we have the cumulative number of arthropod species or compounds, and here we have the year. And so this top bar we have is the number of species with resistance to insecticides. So you see it's pretty slow, but with the introduction of synthetic organic pesticides, we see this huge kick in the number of resistance cases reported. Similarly, the number of insecticides that have reported cases against them takes off. So ultimately, by 2014, which is the most recent numbers I could find, there's nearly 600 insect species that have resistance to at least one insecticide. There's over 325 insecticides with resistance reported for them, and 5 different Bt genes are known to have resistance against them. So this is a big problem. There's a ton of insects out there with resistance. There's a ton of insecticides that don't work anymore, and these are all chemicals that we can't use anymore. At least not in those specific situations. So a huge loss in potential tools. So, how does resistance actually happen? How does resistance come about in insect populations? Well, from an individual level, what resistance is essentially it's when an individual in a pest population evolves a heritable trait that allows them to tolerate or avoid factors that are lethal to the majority of individuals in a normal population. So that's a long description, that's a long definition, but it really hammers on the key points. And ultimately resulting in a lack of expected control when following the label instructions. So the main points here, though, are that resistance occurs at the individual level, that individual insects are resistant, and so when you get enough of them that are resistant in a population, you start to see control failures. Resistance is an evolved trait. So it happens at the genetic level. You need to have some gene that mutates to provide resistance to the individual insect, and that gene needs to be heritable. It needs to be able to be passed on to the insect's offspring. So if you have an individual insect that is resistant to, say, DDT, it doesn't mean much in the grand scale of things if they can't pass that resistance on to their offspring, because you can just wait till they have their offspring and then spray that population. So ultimately, what it comes down to is resistance is created inadvertently by growers through the process of evolutionary selection. It's an evolutionary process, and we just so happen to propagate it accidentally as we attempt to grow our own crops. It's just a byproduct. Seeing some writing and typing, I could wait for a second. Okay. All right. So I just want to do a brief overview of natural selection so we have all the terms fresh in our mind before we start talking about resistance. So, fundamental thing is when we think about resistance, when we think about natural selection and evolution, we're thinking about populations. We're thinking about groups of individuals that live in the same area and breed with one another. So biological populations ultimately are composed of individuals. And those individuals differ from one another in their genotypes. Genotypes being genetic variants that those individuals carry. So for an example here, we've got a population of plants I've just represented with little green blobs. And those individuals have different variants of a particular gene that I'm going to call the tolerance gene, or T. So in genetics, we oftentimes represent gene types or variants of genes with big letters and versus small letters. So here to keep it simple, I've just got homozygotes that are either big T, so dominant for the tolerant trait, or little t's, basically being recessive for the tolerant trait. And at this point, I've got 20 individuals. 12 of them are dominant, so big T, so 60%. And 8 of them are little t, 40%. So we've got individuals that differ in their genotypes, and we measure those as the frequency of those genotypes in the population. So 60 dominant versus 40% recessive. Now, of course, the reason we care about these genotypes is that the genotypes all code for different phenotypes or traits in the population. So, and these traits affect essentially how well an individual survives or reproduces in the environment. So, ultimately, if the big T and the little t, the dominant versus the recessive traits of tolerance, had no effect on the survivability of the population, we wouldn't care. But there's perhaps some ecosystem, some sort of environmental factor, some weather pattern, some predator, something that ultimately makes it so that one of these traits survives better than the other one does. And what we call that is fitness. Essentially, that if individuals have a particular phenotype that is well suited to the environment, they have higher fitness, meaning that they are more likely to survive and reproduce and pass on their genotypes to their offspring. So that's what we call biological fitness. So if you hear things like people saying survival of the fittest, they mean that organisms that are more likely to reproduce are more likely to survive and pass on their genes into the future. Right? So let's imagine a situation here with this hypothetical population. Let's say that tolerance makes the plants tolerant to drought stress. So let's say we have a situation where we've got this field of plants, they're out there, and suddenly we have a couple years like we did just in the last 5 years here in the Central Valley. We're getting a lot less rain. Having the ability to withstand drought stress is going to be much more important. And so you have some individuals die. They die before they reach the reproductive age. They don't pass their genes along. And all of a sudden, our population is looking a little bit different. So that event that we just had, that one in which the individuals die, we would call that a selection event. So selection pressure or selection is any ecological factor that reduces the reproductive success of part of a population. So essentially, anything that happens in the environment that makes it harder for one part of the population to survive than another due to their traits is a selection pressure. So in this case, the selection pressure would have been the drought. It caused it so that organisms with the recessive trait are more likely to survive, whereas those with the dominant trait are less likely to survive. So we had our selection event, and all of a sudden our population's down to 5, 10, 12 individuals, of which 4 have that dominant trait that used to be at 60%. 8, all of the ones with the recessive trait survive because they have a trait that's better suited to droughts. And so all of a sudden our frequency shifted from 60-40 to 33-66 or 33-67. So all of a sudden, one environmental selection event changed the structure of the population at a genetic level pretty fundamentally. So ultimately, the point of this is that genotypes that make an organism well suited to an environment increase in frequency. If your genes make you better fit for the environment, you're more likely to survive, you're more likely to reproduce, you're more likely to have more offspring. And while that may be a small change at a single generation, if you go multiple generations out, that can cause these genes to increase substantially in their frequency. One thing to keep in mind, though, is that no particular gene is fundamentally better than another gene. It's all based on the environmental conditions. If it were to start raining again, perhaps in conditions that there isn't a drought going on, the big T trait is actually the more fit one. It's the one that allows them to have more offspring. And so then big T would start increasing again. It's all about the current structure of the environment and what genes there are out there. Oh yeah, there's my little example of big T increasing again. All right. So ultimately, what happens when we're applying an insecticide is that we are putting a selection pressure on the insect population that favors the reproduction of resistant individuals, right? Anytime we essentially go out there and apply a pesticide, excuse me, I guess I get ahead of myself. So essentially, right, we have pests in a field and they represent our population. Those pests, those insects have different genotypes. And some of those genotypes just so happen to code for genes that affect how resistant the insect is to the insecticide. So when we spray them with an insecticide, we basically increase the chances that the resistant individuals will reproduce, and we increase the chances that their offspring will be present in the next generation. So we slowly build up more and more resistant offspring every single time we apply. So for example, let's say we have a population of aphids. And aphids in this case, are color-coded based on their resistance to a particular insecticide, with these pink guys with the little minus sign being susceptible to the insecticide, and the green guys with the plus sign being resistant. The general rule is that if an insecticide gets all the way to market, it's because the vast majority of individuals in a population are susceptible. It doesn't make sense to sell an insecticide where you know that there are already insects that it can't kill, right? So generally, this is what we would see, is a small number of individuals are resistant, large number are susceptible. So let's say you go through and you apply a pesticide. So you spray it, it kills off the individuals that are susceptible. Who do you have left? You've got your resistant individuals, right? And all of a sudden, when these resistant individuals go around looking for mates, they're not going to find any susceptible individuals to mate with. They're only going to find one another. So essentially, there's a 100% guarantee that all of their offspring are going to have those resistance genes. And thus, 100% of the insects coming out of those eggs, or in the case of, well, it would still be eggs from aphids, are going to be resistant. So just basically to review, resistant genotypes exist in a population, right? They tend to be, though, at very low frequencies. We're talking usually about thousandths or tens of thousandths of a percent of the population. Insecticide applications kill off non-resistant individuals, and resistant individuals then make up a greater portion of the population, making it more likely that they will mate with one another and increasing the percent resistance in the next generation. So one thing that's important to realize from this is that ultimately resistance is what we call pre-adaptive. So what pre-adaptive means is that the trait already exists in the environment. It has to be inherited from your parents. You can't just have it spontaneously evolve from exposure to an insecticide. So this is something you'll hear sometimes when you talk to people who aren't really familiar with natural selection, aren't really familiar with resistance. There's this general feeling that if you go out and you spray an insect population with an insecticide at a lower than lethal dose, that the insects will just somehow magically become resistant to it. They will somehow all of a sudden mutate and have a gene that gives them resistance. The reality is that doesn't happen. If you spray at a lower rate, there's a higher chance that insects will survive just because they're less likely to get that lethal dose. But the reality is they're not just going to spontaneously become resistant. That gene has to exist in the population to begin with, or else, you're not going to see resistance. Oh, there we go. All right. So how is it that insects actually overcome insecticides? How is it that an insect can be resistant? What is the physiological mechanism that an insect uses? Well, there are three major routes. The first of which, and by far the most common of which, is biochemical resistance. So essentially, what biochemical resistance is the insects evolve some sort of ability to detoxify the insecticide before it reaches the site of action in the insect. So you've got this insecticidal chemical, like a organophosphate, that binds on to acetylcholinesterases, right, and stops them from breaking down acetylcholine. What the insect would do is it would have some sort of chemical, some sort of enzyme, some sort of protein that would basically intercept these organophosphates before they get to the nervous system and break them down so that they can't bind the acetylcholinesterase and it protects the insect. And so a general protein that we see in almost all cases of biochemical resistance is this guy, cytochrome P450. Cytochrome P450 is an oxidase. It is an enzyme that basically takes chemicals and it sort of steals electrons, it restructures their format, and it's really widely used by insects to detoxify chemical compounds. Excuse me. So, for example, in cotton bollworm, this guy, cotton bollworm developed resistance to pyrethroids by basically taking its normal cytochrome P450s and then upregulating the number of them they produced 433 times. So they have 433 times more detoxifiers in their body than a normal insect would. And so by virtue of having that many detoxifiers floating around all the time, they can basically take a direct spritz of pyrethroid and survive. because they're detoxifying it as fast as you're pumping it into them. The next one we're talking about is physiological resistance. Physiological resistance has a really useless description, but it is what it is. It's sort of a catch-all category for essentially any process where the pesticide is not detoxified, but it's somehow removed from the body by one or more physiological ways so that the insect doesn't die. So the basic idea is that there are a lot of different mechanisms the insects can use other than detoxifying the chemical, and we just lump them all into this group that we call physiological. It's kind of frustrating, but the reality is if we went out and looked at each of them individually, it would be a huge list. So one example of this would be pyrethroid and DDT resistance in flies. So essentially, what the flies do is pyrethroids, right, and DDT, they basically take sodium channels in the nerves and they prop the door open, right? So sodium always flows through. The sodium channels can't close. The neurons basically lose their charge and the insects are paralyzed. Well, with the DDT and pyrethroid knockdown resistance, what they did is they changed the shape of the door so that the door is no longer able to be wedged open by DDT or pyrethroids. So DDT and pyrethroids aren't being detoxified, but they can't actually have the effect that they used to because they no longer fit the door on the sodium channels. Another example of physiological resistance would be bed bugs. So bed bugs are a big problem right now. I just got a text from my brother. He sent me a picture. He said, please tell me this isn't bed bug. It was a bed bug. He works at a summer camp. Some counselor had it on him, so that's going to be fun. He's going to have so much fun dealing with that. He's their maintenance guy, so it's his job. Oh, it's okay. He's hiking the Grand Canyon for a week, and I'm here with all of you. No, I really do like this. I like all of you. But it would be cool to get the Grand Canyon. Anyway, so bed bugs. One of the big reasons that bed bugs are a big problem right now is they developed resistance to pyrethroids, which is a major chemical we use inside houses. And one of the ways they did that was that pyrethroids get into the insect by basically going through their exoskeleton. And so resistant bed bugs basically evolved a trait that made their exoskeleton 2.5 times thicker than normal, so that it takes a lot longer for pyrethroids to bleed through, and they tend to degrade by the time they get into the actual insect's body. And so the pyrethroid isn't really being detoxified by the insect, they just have a thicker skin. And so it's just another example of physiological resistance. Then finally, there's behavioral resistance. This would be any instance in which the behavior of the insect changes so that they avoid the insecticide. Sort of the classic example of this is Anopheles gambiae, which is the malaria vector most common throughout Africa and at one time southern Europe. But essentially, in Africa, the attempt to control malaria during the 1950s, 1960s was largely focused around spraying DDT on the inside of houses. These mosquitoes would feed on people, land on the wall to digest the blood and turn it into eggs, and then they would go off somewhere and lay their eggs. So what they would do is they'd spray the walls of DDT, the insect would feed on you, land on the wall, get a dose of DDT, and die. But as it worked out, some of the mosquitoes didn't land on the wall inside the house, they'd fly out and they'd land in, say, a tree, or the outside of the house, or they would go in directly to the water source to lay their eggs. And so what they ended up doing was they selected out all the individuals that land on the walls of houses and selected for all the individuals that travel outside the house. And so the population evolved a behavioral change that they prefer to land outside of houses now. And so it makes it harder to control them with indoor sprays. So just kind of a bizarre little shift. All right. So one thing you see with insects is that insects evolve resistance a lot faster than any other system in agriculture. So here we have the number of pesticide-resistant species versus the year in which they were reported. You can see that insects have, they started evolving resistance faster. They have evolved more resistant species than have plant pathogens or than have weeds. And so it's kind of this question of, well, why is it that insects are so good at evolving resistance in comparison to these other systems? Anyone care to venture a guess?
Multiple generations in a season.
Multiple generations in a season, yeah, that's true. You have a lot more reproduction. So if you select, you're having a lot more of those events where the resistant individuals are mating with one another. Any other guesses?
Migration abilities?
Yeah, migration abilities. So if you select an insect in one field, it's very possible for that insect to migrate into another field where it will be selected again. Unlike a weed, which tends to hang out in the same field. I say it tends to, but almost universally do. I'd be really impressed otherwise. Any other thoughts?
The ability to mate with varying types. Like, because when they travel, they can actually mate with...
Oh, multiple different insects. Yeah. So
plants are usually stuck in one area, in one place.
Yeah, so also because they travel, a resistant insect may be able to mate with multiple different insects in different locations, whereas weeds, you can basically either mate with yourself or with your really close neighbors. So those are all really good examples of why. So the main reason that people oftentimes point at insects is their rapid reproduction. So essentially, insects reproduce quickly, and when they do reproduce, they produce huge generations. Colorado Potato Beetle takes less than a growing season to reach maturity, and they lay 800 eggs. So you're producing a ton of offspring multiple times a year. And because you're producing so many offspring, you're producing a large amount of genetic variation, right? You have a lot of opportunities to recombine genes into different combinations that may be pesticide resistant. There's also a greater chance that you'll have some novel mutation when the offspring is born, so that you will just have some random mutation pop up that makes them resistant. And then I guess I should have put in there that there is always the migration issue, that insects can migrate between fields and spread resistance that way. And finally, the last thing is that insects, more or less, since they first showed up, have been in what we would call a chemical arms race with plants. Insects like to eat plants. Plants don't like to be eaten by insects. So plants evolved chemical defenses to kill insects. We have things like nicotine, caffeine, all the chemicals that make plants fun essentially were chemicals that at one time were designed to kill insects. Insects eventually decided they still wanted to eat plants, and so they evolved an ability to overcome those chemical defenses. Plants come up with new chemical defenses, and essentially it is an arms race, essentially. Plants become more well-defended, insects come up with new ways to overcome those defenses. So if we build all of our insecticides off of plant-based chemicals, like neonicotinoids, pyrethroids are based off of pyrethrum, we're essentially throwing the same chemical compounds at the insect as they've already developed the ability to get resistance against. So we're essentially just using the same tools the plants have been using, and we're just accelerating the arms race. We essentially developed the nuclear option, and insects are responding. All right, so general rules of thumb about how quickly resistance evolves. 2 of them. The first of which is that the greater the selection pressure, the faster the rate of evolution. So essentially by that I mean, the more insects that you kill with an insecticide spray, the faster resistance will evolve in the system. And that's because selection pressure essentially reduces the chances that an unfavored genotype will make it into the next generation. And by consequence of that, it increases the chance that a resistance one will get passed on. Yeah, and so the general rule here is that if you have a pest management tactic that has a great mortality, it kills nearly 100% of the insects in the system, these are the ones that are going to be effective for the very shortest amount of time, because you're cutting out almost all of your susceptible insects and you're leaving nothing but the resistant ones behind. So, oh geez, that did not time out well. All right. Oh no, this does work out. I just forgot to fix the animation. All right. So, let's say we have a hypothetical population of insects here represented by these little circles. Green circles with the big R's are susceptible, whereas the red circles with the little r's, these are resistant individuals. So let's say we apply a pesticide that has a 95% mortality rate, meaning it kills 95% of all susceptible insects that it comes into contact with. So we apply it, and ultimately, the post-spray population looks like this. We killed 95%, which comes out to about 46 of the susceptibles, and we end up with 2 of each type, 2 resistants, 2 susceptibles. So ultimately, we went from having what was a 4% resistance rate to having a 50% resistance rate right off the bat. If we assume that these proportions are maintained in the next generation of insects, we would again expect 50% of the population to be resistant. So the next time you come out and spray with this chemical again, 50% of the population won't die. So let's flip that. Let's say you apply an insecticide that has 70% mortality instead of 95. 70% of the, I really should have fixed that. Oh, well, 70% of the 46 is going to leave 30% behind, which comes out to 3, 9, 12, 14 susceptibles, meaning that almost 88% of the population is susceptible, only 12.5% is going to be resistant. And as such, we essentially have a population that is more broadly susceptible than the one where we selected against 95% of them. Yeah, I really screwed these up. Oh, well. That's too bad. Similarly, the more frequently that you apply a selection pressure, the faster evolution will evolve, or resistance will evolve, excuse me. So essentially, looking at that same situation as before with the 70% selection, if we select against the population, we end up with that sort of 12.5% resistance. So let's sort of project this ahead and see what the next generation would look like, where if it was 12.5% resistant. We end up with 6 resistant individuals versus 44 that are susceptible. If we were to apply another 70% spray to this, we end up with 6 individuals of the resistant, left with, let's see, that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 13 susceptibles. And so ultimately, main point being that after 2 selection events, we went from 4 to 12.5% resistant to 31.6% resistance. Every time we apply, we increase the level of resistance every single time. So basically, the rule of thumb is that applying insecticides repeatedly during the growing season will increase the rate of resistance evolution, especially if you're applying the same insecticide or an insecticide with the same mode of action. And also that insecticides with higher mortality will increase the rate of resistance evolution. So oftentimes it's the best insecticides we have that are the ones we lose the quickest. All right. So before we go then, what I'd like to just highlight are some of the major types of resistance we see in agriculture. And so the most common one that we see is the one that we've talked about already, which would be resistance to chemical insecticides, especially synthetic organics. These are chemicals that impose a very high mortality, so it makes sense that we would see the resistance evolve against these the very fastest. They're the ones that leave the most resistant individuals behind. They are also relatively easy to evolve resistance to, like I talked about before. They are based off of plant chemical technology, so they're the ones that are most susceptible to the insect, to the insect's sort of history in the arms race with plants. Excuse me. And they're also widely used across landscapes. So insects tend to be exposed to these chemicals all over the place, not just in one field, but maybe in the next field they move to. And so lots of exposure, high mortality, evolutionary history, these are all things that make resistance evolve really, really fast. I'll leave that up for just a second. All right. Another thing that we've seen resistance against are the IGRs, the insect growth regulators we talked about last time. These are the chemicals that are designed to mimic insect hormones. When these were released, people basically said, the best thing about IGRs is there will never be resistance against these. Because the assumption was, if we have something that mimics an insect's hormone, the insect can't develop resistance against it without fundamentally changing the way they regulate their hormones, without fundamentally changing their physiology. It's like sort of assuming that if you could kill humans or, sterilize them by pumping them so full of testosterone, that they couldn't reproduce anymore, the only way we could evolve resistance to it is if we basically fundamentally changed our body chemistry so we no longer needed testosterone. Well, the thing they didn't account for was the fact that hormones are essentially an important part of insect development, which means that not only do insects need to use them, but insects also have all the tools they need to increase or decrease the levels of the hormones in their body. They have a lot of natural machinery to make sure that hormones only show up exactly when they need to. And so essentially what insects did was they evolved the ability to change when those factors that control their hormones are turned on and off, so that if they get a huge dose of a IGR, they have the ability to decrease the amount of natural hormone in their system so the IGR just sort of behaves like their natural hormone would. So we do see resistance in these. It tends to be weaker resistance than the conventionals, but it's enough that the insects survive and they reproduce, and it makes IGRs less effective. Resistance to insect-resistant plants. So we didn't touch on these in great detail, but there are plants that have natural defenses against insects. Either they are bred conventionally to have that, or they are genetically modified plants with things like Bt genes. So naturally, it's the exact same situation as a conventional insecticide, in the sense that you have some sort of selection pressure, which would be the resistance in the plant, the Bt gene in the plant, and you're selecting for individuals that can actually feed on those plants. So an example of resistance evolving in this sort of population would be grape phylloxera. If you remember grape phylloxera, they figured out that if you graft American rootstock onto European vines, it provides resistance to the phylloxera. And that's sort of half the story. The reality is that resistant phylloxera that can feed on the American roots are known since 1916. So they've been around. They just tend to be in wild populations, not necessarily in conventional fields. Excuse me. And have just recently been found in California in the 1980s. So these things happen over time. And also there are 4 insects that are known with resistance to GMOs expressing Bt toxins. Earworms, armyworms, stalk borers, and corn rootworms. These 4 guys right here. So we do see resistance building up against these naturally or genetically modified plants that are resistant. All right, here's a weird case. This one's kind of fun. Resistance to natural enemies. So this is uncommon, but it is known to occur, that essentially, if there's any sort of selection pressure, insects can evolve resistance against it. In this case, something like the spruce budworm, this guy right here. Spruce budworms, when they reach outbreak populations, oftentimes end up being controlled by viruses. You'll have some virus that sweep through the population. They all end up hanging from the tree branches like this, sort of dropping virus-infested hemolymph all over other spruce budworms. Turns out that there are some populations that have evolved resistance to this. So you can introduce the virus and they don't die. They have a natural sort of defense against it. There are, as I said before, there's resistance to Bt insecticides, which do count as natural enemies to a certain extent. But this one's really cool, P aphids. So P aphids, they've evolved a partnership with this bacteria called Hamiltonella defensa. It's a bacteria that lives inside their gut and as well as inside their hemolymph. And what it does is it provides defense against parasitic wasps as well as lady beetles. So essentially, if a parasitic wasp comes along and stings a P aphid that is carrying around this defensive bacteria, when the egg hatches and the larva comes out, the bacteria will attack it. They will infect the larva, they will encapsulate it so it can't actually eat the aphid from the inside, and will ultimately kill it. It's not a 100% thing, it doesn't always work, but individuals carrying Hamiltonella defensa do survive parasitic wasp stings at a much higher rate than those without. Similarly, it was found that lady beetles that fed on aphids carrying around this defensa had a significantly higher mortality rate. So populations with lots of Hamiltonella defensa in them tended to be more resistant to lady beetles. So just really kind of a bizarre system, kind of interesting. Not something you see very often, but it does occur. And then finally, last one before we go, is resistance to crop rotation. So crop rotation, if you remember, just kills pests by depriving them of their host for an extended period of time. And it's a system that's really hard to evolve resistance to because it's really hard to survive not having a host. If you don't have anything to eat, you just can't survive. So a good example of this would be in the case with northern corn rootworm. These guys are found throughout the northern US, where what they do is they feed on the root of corn The adults emerge, they feed on the silks of the corn, and then they lay their eggs in the soil so that the larvae can feed on the roots again. It's just this really simple life cycle. For a long time, the way we controlled this was you rotated corn with soybeans. These guys can't feed on soybean roots. There's nothing above ground for them to feed on either. So the larvae would hatch, they'd crawl around, they wouldn't find any corn roots, they would die. However, in the 1980s, there was a rootworm in Iowa that evolved a 2-year life cycle. So it would lay its eggs, the eggs would overwinter for a full year. They would never hatch, and then when the second spring came, they would hatch when the corn came back. So essentially, you had this nice sort of system where you'd lay your eggs, you wait a year, and then you hatch and you got corn. This one never really took off. It was kind of a little problem, but the gene didn't provide really enough fitness to take off. The real problem was in the 1990s, there was an Illinois variant of the rootworm, and what it developed was the ability to basically have a different host preference for laying eggs. No longer did they like to lay their eggs in cornfields, they like to lay their eggs in soybean fields. So essentially, you'd have all these rootworms feeding on the corn, and then they'd fly over to the neighboring soybean field, lay their eggs in there, and of course, next year when the corn gets planted in that soybean field, you've already got an inoculation of all of these eggs there waiting to feed on the corn.
So is that like an example of behavior change or whatever?
That would be a great example of behavior change. So this would be an example of one behavior change in the Illinois population. How about this one? Physiological, right? They changed some aspect of their physiology, but they didn't actually detoxify well, there's nothing to be detoxified, it's crop rotation.